Hi guys, Mrs. H here. We're going to go ahead and take a look at DNA and RNA structures. So when we're looking at this, remember our key vocabulary that we looked at yesterday and what we want to be able to do. We want to be able to explain the structure and function of DNA and RNA, understand what a nucleotide is, and then compare and contrast the DNA and RNA. So, what is a nucleic acid? As a review, remember those nucleic acids are a macromolecule. Those macromolecules or biological molecules are one of the four components that make up all living things. When we're looking at nucleic acids, we're looking at primarily DNA, and DNA being the carrier of our genetic code and for heredity. We do know that in some cases that some objects or some things, not objects, some things do have RNA as their genetic code, but that's very rare. Most organisms do use DNA. And we know that DNA is like the instruction book. It's the recipe for making you, and making all living things what they are. And so those instructions are embedded into that DNA molecule. And RNA is kind of the helper that helps DNA out. So generally speaking, we say that DNA is a double helix, and DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And both of those have nucleotides as their monomer to construct. So the monomer, that is that brick that makes a building. So the monomer is the brick that makes the biochemical compound. Both of these, notice, have nucleic acid at the end. That's where the Na comes from. And then the D and the R, the deoxyribo and the ribo, comes from the difference between those two structures. And so when we look at the roles of DNA and RNA in our body, DNA is, like I was saying before, it is the instructions. It's the directions that make up everything about you. And so if you want to perform any task as a living organism, the instruction for doing that task or for making what needs to be made is in the DNA strand. It's encoded into the DNA strand. Where once in a while we'll see RNA as the genetic molecule, and that's usually in a virus. And there's some debate on whether or not a virus is really considered a living thing or not. It does behave like a living thing, but the discrepancy on whether or not a virus is a living thing is whether or not it can reproduce on its own. It can reproduce, but it needs you or it needs another living thing to do that. And so it's kind of one of those debates on whether or not we even say RNA can be a genetic material, because is it a living thing or not? So in our eukaryotic cells, the DNA molecule is in a structure called a chromosome. We only see chromosomes in our eukaryotic cells. We won't see the chromosome in, say, bacteria, which is prokaryote. So what exactly is a chromosome? A chromosome we usually think of as that X-shaped structure. We only see chromosomes in an organism when it's about ready to divide. And basically, DNA is really, really big, well, not big, long. It's extremely long. And so we need to condense this DNA strand when we're getting ready to divide our cell. And so we need to make it easily transportable. So if I'm going to be packing on vacation, I'm going to put all my luggage into a trunk. That's the case that I can transport and move my clothes around. Well, chromosome is a structure that we can move DNA more efficiently around. And so what a chromosome really is, it's a long strand of DNA that will actually wrap itself around a histone, and histone is just a little protein, a circular rod-shaped structure. You can see them right here, and they wrap up around those, and then these create these clusters, and then these clusters actually will start to supercoil, and then it'll coil again and coil again and coil again. So this right here, this X-shaped structure, is what we know of as the chromosome, where this structure, this twisted staircase-looking structure, that double helix, is what we refer to as DNA. So the difference between these two is DNA does not have any of these histones, and it's not in this supercoiled position. So when DNA wraps itself around these histones and puts itself into this supercoiled position, it becomes known as a chromosome. And a lot of times these words are interchangeable in common everyday language, but they are definitely different in a biological context. So roles of DNA and RNA in living things. So in eukaryote, chromosomes are found in the nucleus, in that double-bound membrane, where the DNA is not found in a chromosome structure, and it's not even found in a nucleus. And so those are some big differences between our eukaryotic cells and our prokaryote cells in terms of DNA. Because bacteria is so small, there's not a lot to it. There's actually not a lot of DNA. They're a much smaller amount of DNA in bacteria than there are in eukaryotic cells. But we will find the DNA will stay in a general area within the prokaryotic cell, even though it doesn't have a membrane that traps it there like the eukaryotic cell does. The eukaryotic cell has a safe, if you would, for the DNA, so that it's protected and it doesn't risk of getting damaged and doesn't get risk of losing the DNA or the strands of DNA. So it's housed within that nucleus. And because the DNA is housed within the nucleus in our eukaryotic cell, we need RNA to help the DNA perform this job of creating an organism and allowing the organism to function. And so DNA contains thousands and thousands of genes or instructions that we synthesize into proteins. So it's like taking our recipe and getting all our equipment to make our cookies. So we get our DNA, we get our RNA, so that we can make an organism or perform a task or perform one of life's processes. That's how it kind of the sequence of events. So the RNA is the link between the DNA instruction and the protein that we actually make. And proteins are what make every organism you. So whether it's being able to reproduce a new cell or reproduce as an organism or maintain homeostasis or obtain and use energy, we need proteins to do all these things. So the connection is we have the instructions to do all those things and we make it into protein so that we can perform those life processes. That sequence of events is actually known as the central dogma. that includes two processes, which is transcription and translation. We will elaborate on this part in the coming weeks. Today, we're specifically just looking at the roles of DNA and RNA and what those structures are. So because DNA has to stay in the nucleus and proteins are made in the ribosomes, we need to use the RNA to link. It's the link between those two places, between the nucleus and between the ribosomes. So that mRNA is a helper to allow those processes to happen. So what do these structures look like? So our DNA and RNA molecules are polymers, poly meaning many, and they have many of these nucleotides, which is the monomer that makes up this up. And so because we have many nucleotides, it's sometimes called a polynucleotide. And so what do these nucleotides compose of? Well, we have a nitrogen base, we have a sugar, and then we have a phosphate group. And typically speaking, we use a circle to represent the phosphate group. We use this hexagon as to represent the sugar, and then we use this. Sometimes you'll see a triangle, but we'll see either a hexagon by itself or a hexagon with a pentagon attached to it. And all of these three things together make up that nucleotide. And so this nucleotide then is repeated. Here's one, here's one, here's one, here's one. So notice we've got multiple, we've got many of those nucleotides creating that polynucleotide. And so let's look at this structure a little more closely in what these nucleotides are consisted of. So like I said before, notice this two-ring structure. This represents, and notice also the triangle, or not triangle, rectangle. This represents the base. There is in DNA 4 different bases that can be found in DNA: adenine, thymine, guanine, and cytosine. The sugar is deoxyribose. Deoxy, hear the word deoxyribose, deoxyribonucleic acid. So the name DNA comes from the sugar found in DNA, where RNA or ribonucleic acid has ribose as a sugar. Also to note that notice the sugar has that OSE ending. And so there are one of two types of sugars found in a nucleotide, and it is based on what kind of nucleotide is it. Is it a DNA nucleotide or is it a RNA nucleotide? And there is no difference in the phosphate between a DNA and RNA. So again, this is the same, the phosphate group. We can have either deoxyribose or ribose for a sugar. And then for DNA, there's one of four: adenine, guanine, cytosine, and thymine, or in RNA, adenine, guanine, cytosine, or uracil. Uracil is different than the DNA molecule. Oh, and this is what I just said. I'm sorry. I forgot to advance my slide. So this is what I was referring to. So these are those bases that are different. Notice they even look different. So our cytosine and thymine and uracil, they have one ring, where our purine, which is adenine and guanine, has two rings. And so cytosine, thymine, adenine, guanine, those four again are found in the DNA, where these four over here, the cytosine, uracil, adenine, and guanine are found in those RNA molecules. And because these three have one ring, we call them pyrimidines. And then because these have two rings, we call them purines. This also, is used to look at the direction. So when we're looking at the nucleotide, is it right side up or is it upside down? So how can we tell if it's right side up or upside down? We actually look at the sugar molecule, and that's going to be important when we get into replication, when we get into transcription, translation, which are processes that we'll look at in this unit. So how do we know what direction these are facing? We actually look at the sugar. And so basically what we're saying is the nucleotide upside down or is it right side up? And so we look at the sugar to be able to do that. Another difference another thing that we look at with the sugar is we look at what's the difference between the deoxyribose and the ribose. And if you think about the word D means to get rid of, oxy is oxygen. And so the difference between the deoxyribose and the ribose is this is missing a sugar. There's one less sugar in this molecule, and it's actually right here. So do you see how this has an OH? It has an oxygen over here. There's no oxygen there. And so the sugar molecule is actually really important to the structure. It tells us whether it's right side up or upside down. And then it also allows us to look at the difference in our DNA molecule and our RNA molecule. So how do we know whether or not it's upside down or right side up? We actually number our carbons. And so each one of these points on the sugar molecule is a carbon. And so we number them. So this is carbon 1, 2, 3, 4, and then there's a carbon off here, like tagging off on the hexagon. And so we count the carbons to know whether it's upside down or right side up. Is the 5, right? The 5 up here, is it facing up, or is that little tag flipped downwards? So we just flip this. Is this flipped upside down? And so that's how we can tell the direction of those sugars, is based on counting those sugars around the rings and where is that little extra tag on that molecule ring. So let's take now a look at the phosphate group. So a nucleotide by itself will actually just be, will actually have 3 phosphate groups attached to it. So if this nucleotide is not attached to a structure, it will actually have 3 phosphates. So sometimes it's referred to as a triphosphate because of the 3 here at triphosphate. And this is important because of how we actually add nucleotides to our molecules. I'm not going to go ahead and spend a whole lot of time on this, but I just wanted to reference this here, that by themselves they'll have these 3 these 3 phosphates. So how do we get this chain? What's the direction? And this is specifically referring to our DNA. And so if you look on this side of the DNA, So on this side of the DNA, notice the orientation of this sugar, okay? And so it looks like it's upside down. And so this 5, right? The 5, 1, 2, 3, 4, 5, the 5 prime end is on the bottom. And so on the top is this one right here is the 3. And so what we say is this strand is in the 3 to 5 direction, because the 3 the 3 carbon, the number 3 carbon is right here, and the number 5 carbon is right here on that outer edge or the backbone. And so this is the 3 to 5 strand of the nucleotide, this DNA nucleotide. And then on this side, notice it's in the other direction. And so the 5, that carbon hanging out by itself is right here. And so then this is 4 and this is 3. So this side of the strand is called the 5 to 3 train. As a result of this, because one side of the molecule is facing this direction and the other is this, we call this anti-parallel. So these structures are on DNA is anti-parallel. And so how do we attach these nucleotides together? Remember, we've talked about this process before of losing water when we make these. So we're going to lose a hydrogen here and we're going to lose a hydrogen here to attach these 2 nucleotides together. So we again call this a hydrolysis process. Okay, so when we're looking at this DNA strand and when those 2 strands start to form and are bonded together, they're going to actually naturally twist and create this double helix structure. Because of the bonds and the orientation on the bonds, they're going to want to, they're not going to want to stay straight. They're going to start curving and start bending. The other thing to note is when this happens, these on this model, the strings on the outside, this is where the phosphate and the sugar are found. And then inside here, this is where the bases are found. So oftentimes we say the ladders of the DNA molecule is composed of alternating sugars and phosphates, and then the steps of the ladder are where the bases are found. Now these bases, so how do these 2 sides hold together? They're actually held together by hydrogen bonds. And so if we look at this a little more closely, we're going to look at those bases. These bases actually bond together very specifically. Adenine and thymine are going to bond together. So notice adenine has 2 rings and it's bonded together with a thymine that only has 1 ring. Where if you look at cytosine and guanine are bonded together and we have a 2 ring with a 1 ring. And so the lengths of the steps, if you will, end up being the same length. And so you don't have like this whapper-jawed ladder. Your ladders are equal distance all the way up and down and not in and out and in and out. That would not be a very stable ladder, just as it wouldn't be a very stable molecule. So what else do we notice happening with these bonds? Well, 1, the adenine is always with thymine, cytosine is always with guanine. But there's also something else to note in particular. Notice the number of bonds. Notice adenine and thymine are bonded by 2 hydrogen bonds, where our guanine and cytosine are actually going to be bonded with 3 hydrogen bonds. And that's important in the molecular structure. Adenine and thymine can't bond with 3. It will only bond with 2, where cytosine and guanine need to have 3 bonds. And that makes it a very secure and tight structure. It makes it very stable, even though hydrogen bonds are a weak bond. Where our bonds between within our adenine and within our sugar and within our phosphate, these are covalent bonds and they're very strong bonds. Where hydrogen bonds are weaker bonds, but because there's so many of them, it does make it for a stable, predictable pattern, if you would. So again, if we're looking at these, we're looking at these two strands, and we discussed this before. This is your 5N and your 3N, and we can really see it more closely here. And so these two strands, even though they're bonded adenine and thymine, the one strand has to flip upside down in order for the two structures, for those two bases to come meet. And so that's why these being anti-parallel and one being in one direction and the other in the other direction, that 5' to 3' and the 3' to 5' is so important. It allows those bases to bond together. And again, creating an anti-parallel structure. So what makes RNA different? Well, we've already talked about the ribose, and we've also talked about the uracil. But the one really big thing that's different is that the DNA, we say, is a double helix, where our RNA is actually a single helix. So again, if we're looking at our differences, we have that thiamine and uracil. There's the base difference. We have a sugar difference. That's 2. And then also the double helix compared to a single helix. So there's 3 distinctive differences in structure between that DNA and RNA. So another difference is DNA is just DNA. There's only one kind of DNA. There's actually 3 different kinds of RNA. The first one being messenger RNA, and messenger RNA is the copy of DNA. So I don't want to take my information out of my nucleus. So I need to make a copy of whatever instructions that I need, and I make that copy in a strand of mRNA. So DNA is going to stay in the nucleus, and mRNA is going to leave the nucleus after it's been made from the DNA strand. Messenger RNA will then travel to the ribosome and meet up with ribosomal RNA. Ribosomal RNA is what makes up the ribosome. It's a component of the ribosome, which also has enzymes on it so that a chemical reaction can occur in the site. And that chemical reaction is creating the proteins. Another difference that we see, oops, what did I do here? Oh, this is not right. This should be titled, sorry, not messenger RNA. This should be called transfer RNA. I will make sure that is changed on your notes. But tRNA or transfer RNA is actually has this clover shaped structure. So it's not that long strand or the ribosomal clump. This is actually a clover shaped. And what the transfer RNA does, it actually brings to the ribosome the parts to make the protein. So it brings amino acids to the ribosome so that we're able to make a protein. So each one of these has a distinctive difference in our in its structure and in its function. So tRNA, mRNA, rRNA, they have different shapes and they have different functions, but all of them are helping to make a protein, to make you. So here's a summary of what we just went over. So go ahead and take a minute to read this. And if you have any questions, please make sure you're reaching out to me and asking for help. Bye, guys.